presentation. And the last session for today's virtual event, we have the session on using genomic tools for botanical and natural ingredient identification, that is an industry perspective. And for that, we have Dr. Liu from application scientist, Thermo Fisher Scientific. And here I would like to introduce Dr. Liu as he's a senior analytical scientist at Herbalife Nutrition. And Mr. Liu holds a PhD in experimental pathology from the University of South California and received a medical degree from Peking University. With over 15 years of experience in the field of molecular application, he joined the dietary supplement and food industry to work on DNA-based authentication methods since 2016. His work involves characterization of DNA in a botanical materials, developments and validation of DNA methods for the identification of both raw herbal materials and processed botanical extracts. Implementation and ISO 17025 accreditation of analytical methods for routine botanical qualification. His work has been published in Food Chemistry, Phytoria, and Journal of AOAC International. He plays active roles at AOAC, whereas he shares and promotes analytical solutions by sponsoring multiple AOAC scientific sessions on molecular application and participating food authenticity working group to fight food fraud. In addition to his role at Herbalife and AOAC, he also advances public quality standards by volunteering his time and expertise to co-chair an United States Pharmacopoeia, that is USP project team on botanical library for identification using DNA-based methods. That is 2015 to 2020 cycle. And as a member of USP Botanical Dietary Supplement and Herbal Medicines Expert Committee from 2020 to 2025 cycle. Now I would like to invite on screen Dr. Liu, Senior Analytical Scientist, Herbalife Nutrition.
Good afternoon, Indian colleagues. My name is Zhengfei Lu. I'm a senior analytical scientist from Herbalife Nutrition. In the next 30 minutes, I'm going to talk to you from an industry perspective on using genomic tools for botanical and natural ingredient identification. We will cover specific topics such as genomic tools as a part of a comprehensive approach to botanical and natural ingredient identification. Our experiences on different techniques in the genomic testing toolkit, what we already have, and why NGS is an essential technique. In the end, challenges and solutions of NGS application in botanical and natural ingredient identification will also be discussed. A comprehensive approach to botanical and natural ingredient identification. Current good manufacturing practices require dietary supplement manufacturers to verify their raw material identity before use. Among those raw materials, botanical and natural ingredients belong to a unique category. In terms of market trends, due to the consumer perception that naturally originated botanical ingredients are premium and healthy, botanical ingredients are popular components in dietary supplement and food products. However, with the growing demand, the potential for adulteration or substitution are also on the rise. To detect adulteration of botanical and natural ingredients, fit-for-purpose methods are required. Given the complexity of botanical materials, a combination of analytical procedures is used to define the diverse attributes of material identity for design specificity. People conventionally use morphological and chemical analytical methods for botanical identification. So does Herbalife Nutrition. Visual inspections, either macroscopic or microscopic, are efficient in many cases. For example, cinnamon bark can be easily differentiated from cassia by thin and paper-like texture. However, morphological features are not always unique and well preserved for botanical materials in commerce. Herbalife Nutrition also uses multiple chemistry techniques such as HPTLC, HPLC, LC-MAS, and NMR in its laboratories for botanical material identification. By the way, these are the most frequently used approach for botanical and natural ingredient identification. In this slide, you are looking at chamomile and feverfew flowers. They have little differences in their appearance, and these morphological features are further lost when they are made into powders and extracts. This slide shows the HPTLC method was used to search for specific marker compounds in chamomile in order to differentiate flower extracts derived from these two species. However, there are still challenges. First, for botanical materials from the same species, the composition of these compounds may vary by harvest seasons, geographic locations, storage and processing conditions. Secondly, when you are facing many close species, not all marker compounds used in chemical analytical methods are specific. To understand variations in chemical fingerprints, authenticate specimens from multiple populations need to be collected, and statistical models need to be compiled. It takes time. To resolve complicated phylogeny, additional chemical standards and markers shall be identified and utilized. It puts burden on the budget. Both time and budget impact routine identification practice. Recently, genomic methods merged as identification techniques that complement conventional approaches. Since then, they grow in importance due to the following. Good afternoon, Indian colleagues. My name is Zhengfei Lu. I'm a senior analytical scientist from Herbalife Nutrition. In the next 30 minutes, I'm going to talk to you from an industry perspective on using genomic tools for botanical and natural ingredient identification. We will cover specific topics such as genomic tools as a part of a comprehensive approach to botanical and natural ingredient identification. Our experiences on different techniques in the genomic testing toolkit, what we already have, and why NGS is an essential technique. In the end, challenges and solutions of NGS application in botanical and natural ingredient identification will also be discussed. A comprehensive approach to botanical and natural ingredient identification. Current good manufacturing practices require dietary supplement manufacturers to verify their raw material identity before use. Among those raw materials, botanical and natural ingredients belong to a unique category. In terms of market trends, due to the consumer perception that naturally originated botanical ingredients are premium and healthy, botanical ingredients are popular components in dietary supplement and food products. However, with the growing demand, the potential for adulteration or substitution are also on the rise. To detect adulteration of botanical and natural ingredients, fit-for-purpose methods are required. Given the complexity of botanical materials, a combination of analytical procedures is used to define the diverse attributes of material identity for design specificity. People conventionally use morphological and chemical analytical methods for botanical identification. So does Herbalife Nutrition. Visual inspections, either macroscopic or microscopic, efficient in many cases. For example, cinnamon bark can be easily differentiated from cassia by thin and paper-like texture. However, morphological features are not always unique and well preserved for botanical materials in commons. Herbalife nutrition also uses multiple chemistry techniques such as HPTLC, HPLC, LC-MAS, and NMR in its laboratories for botanical material identification. By the way, these are the most frequently used approach for botanical and natural ingredient identification. In this slide, you are looking at chamomile and feverfew flowers. They have little differences in their appearance, and these morphological features are further lost when they are made into powders and extracts. This slide shows the HPTLC method was used to search for specific marker compounds in chamomile in order to differentiate flower extracts derived from these two species. However, there are still challenges. First, for botanical materials from the same species, the composition of these compounds may vary by harvest seasons, geographic locations, storage and processing conditions. Secondly, when you are facing many close species, not all marker compounds used in chemical analytical methods are specific. To understand variations in chemical fingerprints, authenticate specimens from multiple populations need to be collected, and statistical models need to be compiled. It takes time. To resolve complicated phylogeny, additional chemical standards and markers shall be identified and utilized. It puts burden on the budget. Both time and budget impact routine identification practice. Recently, genomic methods merged as identification techniques that complement conventional approaches. Since then, they grow in importance due to the following two advantages over conventional methods. First is specificity. The morphological features and the phytochemical profiles that evaluated by conventional approaches are the product of plant genome and environmental interaction. Being able to access genomic information provides direct information to confirm the taxonomic identity of herbal materials at species level. Second is stability. Unlike morphological and phytochemical attributes that changes throughout processing stages, genomic information can be retrieved from different forms of herbal material and is not subject to changes from environmental conditions. To achieve better identification specificity and overcome the obstacles of using conventional approaches in routine botanical identification practice, at Herbalife Nutrition, we also combine genomic tests with conventional identification methods for botanical and natural ingredient identification. Now let's move to the second part and talk about how genomic tools are used. Here are a few techniques proposed in the literature. For routine botanical identification practice, DNA barcoding and species-specific PCR are the two most frequent used techniques. DNA barcoding. DNA barcoding is a method of species identification using a short section of DNA from a specific gene or region. The assumption is that by comparison with a reference library of such DNA sequence, an individual sequence from a sample can be used to uniquely identify an organism to species level. In the same way that a supermarket scanner uses the familiar black stripes of barcodes to identify an item in its stock against its reference database. Does it work? Yes, it works quite efficiently. In a validation study involving 4,500 medicinal plant species, a single barcode ITS2 is able to achieve around 93% correct species identification without making mistakes. This is easy to know that DNA barcoding does not only identify the botanical material, which fulfills the regulatory requirement for botanical species identification, but also examines the other species, which further reduce the risks for commercial adulteration to protect consumers and companies. Obstacles that prevent the wide application of this technique are the lack of curated database and validated method. As you know, public database contains DNA sequences submitted by researchers from different countries. Some of the items may contain wrong identity information. Using such database as reference in quality control is risky. In addition, for industry application, standard operating procedures needed to be established and validated. To provide a solution, Herbalife Nutrition built a curated database using botanical reference materials for common botanicals in dietary supplement industry. Developed and validated the method and shared with the industry through publication. This is our validated method published in the Journal of AOAC International. This method has also been ISO 17025 accredited to meet industry standard. Right now, Herbalife Nutrition Botanical Extraction Facility is using this method to test every lot of starting materials to ensure their correct identity. In this example, DNA barcode sequences are retrieved from Phoenix ginseng roots. These barcodes were compared to a curated database, which does not only have the targeted species, but also contain other close species. For Phoenix ginseng, there are other botanicals with ginseng in their common names, such as American ginseng, Tianqi ginseng, and Siberia ginseng. At data analysis stage, the barcode collected from samples will be compared to all these species, and the results were ranked based on the percentage of identity and length. And the sample was identified as Phoenix ginseng, as it is the best hit in the search. DNA barcoding is a powerful method, but it has its own limitations. First, because it uses universal primers to target amplicons, there is a size limitation. Since traditional DNA barcodes range from 400 to 800 base pairs, DNA barcoding only works for fresh and lightly processed botanical materials, where high-quality DNA can be extracted. Second, Sanger method only sequences a single DNA fragment at a time. For botanical materials in powder form, it may contain a mixture of botanicals. In this case, DNA from all botanical ingredients will be amplified by the same set of universal primer, resulting in unreadable sequencing profiles from the sample. In contrast, species-specific PCR provides a good alternative to overcome these limitations. Species-specific PCR, which designed and validated to amplify short DNA fragments, usually less than 200 base pairs, may have advantages to address species identification in heavily processed botanical materials containing partially degraded DNA, such as extracts. In addition, these methods are expected to be specific to the target article. In other words, false positive results will not be generated in the presence of foreign substance. At Herbalife Nutrition, we also have developed and validated PCR methods for multiple botanical species, including chamomile, papaya, cinnamon, blueberries, and so on. This slide shows our efforts towards this direction. To demonstrate this concept, we took German chamomile as an example, developed species-specific PCR to identify German chamomile and detect other chamomile species, validated the assay in powders and extracts. In addition, we also co-authored another article that lists the recommendations on the validation of species-specific PCR. However, species-specific PCR is neither the ultimate solution, as species-specific PCR achieves its specificity by amplifying specific sequence from the targeted species of interest, while not amplifying non-targeted species. Additional tests can be designed for the detection of common adulterants, like what we did in the chamomile assay, but the test is limited by prior knowledge. Species-specific PCR can be used as a rapid test to deliver answers, but the cost of developing and validating each assay at a species level may outweigh its benefits. While we're looking for a new solution, massive parallel sequencing, also known as next-generation sequencing, NGS, emerged as a promising technique. Now let's see an example. This material was claimed as parsley leaf flakes because it is claimed as parsley leaf, so it was analyzed against the parsley leaf botanical reference materials by HPLC and HPTLC method. HPLC showed a perfect match between sample and BLA. HPTLC showed some band intensity differences, but the majority of the bands match. I am not a chemist, but I will probably release a sample based on the matching chemical profile between the sample and the reference. However, when we perform DNA barcoding, double peaks are presented across the whole trace file. This observation has big impact on decision making, because clean trace file is expected from pure parsley material. Where does these double peaks come from? Could this material contain something else? As we discussed, PCR cannot solve the issue because the prior knowledge is required. If you don't know your target, how can you develop a method to detect? This is where NGS will come in handy. Before we jump into the solution, let's talk about the concept of DNA meta-barcoding. DNA meta-barcoding is a special case of barcoding applying to the samples that contain more than one organism. Meta-barcoding utilizes the same reference database as barcoding, but allows identification of taxia from mixed samples by using high-throughput sequencing methods. This is exactly what we needed. High-throughput sequencing allows us to examine the barcode sequences at a single molecule level, instead of sequencing the barcode regions one by one, like Sanger sequencing method did. We can pull multiple barcodes in the sample run in the same run for better specificity. After extracting genomic DNA from sample, 18 amplicons that originated from the following barcodes are enriched. Most of them are between 200 to 400 base pairs, which is ideal range for NGS. These amplicons were sequenced on NGS platform at a 300 base pair end and mapped to in-house meta-barcode database to identify their botanical origin. The occurrence of all mapped reads were counted and used to plot this pie chart. It turns out the material does contain parsley. 11% of the mapped reads were assigned to parsley, and they come from 10 applicants. However, most of the reads, 87%, were mapped to celery. This result reveals the sample might be adulterated by celery. For your information, the cost of celery leaf is only a quarter of parsley leaf on the market. With the information provided by NGS and only by NGS, celery leaf was added in the chemical analysis. Now, small variations between samples and BRM started to make sense. The material was eventually rejected because it was adulterated with celery. The way to use NGS goes beyond meta-barcoding. High-throughput sequencing allows one to sequence the whole genome instead of only focus on the barcodes. Then, how can whole genome sequencing using NGS help us to fight food fraud? Barcoding methods achieve species identification for most of the botanical materials. However, due to the limited length, the available code barcodes may not always be informative between close species. For example, co-DNA barcodes lack the power to differentiate igneous species. The work cited here were published by Dr. Handy's lab at US FDA. For those who are not familiar with genomics, I'm talking about the phylogenetic tree you see in the bottom and the middle right. This type of tree is usually built to show the similarities and differences between different species in terms of their genetic characteristics. In the tree, one should look for branches in the red circle, called a clade, which suggests the species in that branch is different from the others. You can see that in addition to the two red circles, there is not much separation. This indicates even a combination of DNA barcodes lacks the power to differentiate igneous species. Why? As I said, length is limited. The variation within these code barcodes is not enough to tell us the differences with confidence. A wider region needs to be sequenced. Whole genome sequencing extends the length of sequence regions by covering the whole organism genome, so variations across the genome can be analyzed, and this can only be done by NGS. In their study, whole chloroplast genome were assembled from whole genome sequencing data for comparison. Chloroplast genome is part of the whole genome. It has high copy number in plant cells, so high-quality data can be easily extracted even in very shallow whole genome sequencing data for comparison. The variations we are looking for are called single nucleotide polymorphism, SNPs. For those who are not familiar, when you align a DNA sequence from different species, SNPs are the positions that show different nucleotides, like the A and G you see here. They found that single nucleotide polymorphism between any two igneous chloroplast genome ranged from 181 to 910 and can provide robust data for unambiguous species delimitation among 9 igneous species. Now, in the phylogeny tree, you get all the branches. From industry perspective, We are interested to know if this finding can help us to identify Echinacea species in real life. Following the same SNP idea, we used whole genome sequencing on 28 previously authenticated Echinacea species samples. All the samples are sequenced in one NGS run at 2 by 75 base pairs, resulting in less than 1 million reads per sample. 124 high-quality SNPs were extracted and underwent a principal component analysis. In the figure on the right slide, you can see the circles in different color represent samples of different identity. You can see that the chloroplast genome SNP PCA analysis can clearly differentiate 3 different Echinacea species. This is a good example to demonstrate how genome sequencing solves taxonomic identification that once is difficult to be achieved by other genomic techniques. Challenges and solutions of using NGS as a quality control tool. As you can see, NGS have a lot to offer. That's why NGS was selected as the best method fitting for purpose in molecular application by the Food Authenticity Method Program of AOAC International. However, compared to other established genomic methods, NGS is still a new branch and used mostly for research purposes. In order to facilitate the application of NGS methods in fighting food fraud, food and dietary supplement industry is looking for validated methods that meet designed performance requirements. To provide guidelines for method submission, AOAC stakeholders formed a molecular application working group to draft standard method performance requirement, also known as SMPR, for untargeted NGS assay. Under Daniel Soy's leadership, we already developed a first draft guideline for method validation. The SMPR draft specified the analytical technique is NGS and its application scope to botanicals and spices. It also lists performance characteristics of potential methods, such as targeted DNA region, primer selection and design, database content, algorithm concept, and evaluation of non-targeted DNA sequence. In addition, recommendations on matrices study for single and multi-lab validation were also given. Using this opportunity, I want to thank all the stakeholders. Your participation and commitment helped us reach where we are today. I also want to specifically thank Daniel from Thermo Fisher Scientific to lead this discussion and draft the SMPR. Before concluding my talk, I want to say this is still unfinished business. This still requires industry collaboration. And AOAC is such a great platform for us to collaborate together. We are looking for input, participation, engagement from our Indian colleagues as well. Thank you.